Okay then, as you guys know, my name is Janhavi, and today I'll be presenting my project characterizing Xyloglucan and CNC interactions, a binding capacity study. And for this project, I was really helped by my mentor, Ariane Fernandez from the University of British Columbia. So before we can really dive into what the project's about, we need to first understand a few key terms. So cellulose is a very abundant carbohydrate polymer found in plant cells, specifically in the plant cell wall. And cellulose nanocrystals is form of cellulose, but in like a crystal form. And to get it in this form, acid hydrolysis is performed, where it takes the long cellulose polymers and cuts them down into shorter polymers and aggregates them. Another term that would be useful to know for this project is hemicellulose, which is a carbohydrate polymer composed of many sugars. And this is also an important part of the cell wall because it interacts with cellulose in the cell wall and has like strengthening properties. And a type of hemicellulose sugar is xyloglucan, which is what we're focusing on in our study, and it's derived from tamarind. So it has been observed that xyloglucan adsorbs onto cellulose nanocrystals, and adsorption is a process by which ions, atoms, or molecules just stick onto the surface of solid material. It's important to note that adsorption is different from absorption, and so that's a key difference. And the objective of our project is to study the interactions between xyloglucan and CNC and quantify them. So in the first picture you can see on the left, this is what adsorption looks like. As you can see, the xyloglucan particles, they're not being absorbed, but rather adsorbed, where they're just sticking to the surface. And on the right, that's a picture of cellulose nanocrystals, if in case you're wondering what they look like. And as you can see, they're like rod-like, crystal-like structures. And yeah. So in our study, we used the Orsinol method. And this is basically, it's a procedure where orsinol, the compound, it's a compound which reacts with xyloglucan, forms a cherry red solution, and the cherry red color can then be passed through the spectrophotometer and measure unknown concentrations of xyloglucan. So it's a really helpful, useful method in our project. So here's a review of the methodology, where first we prepare the solutions, then we prepare the samples and centrifuge them, then we perform the Orsinol reaction, and then we use spectrophotometric analysis. So in our experiment, we first prepared our solutions, and here I have listed the solutions we needed for our project. And so our solutions such as sulfuric acid and sodium chloride were first prepared, and our samples, which had differing ratios of xyloglucan to CNC ratios, those were also prepared. As you can see in this big diagram on the bottom, sample 1 versus like sample 10, it has a different final xyloglucan to CNC ratios, and we kind of increased them as our samples went on. And we want to see kind of like how increasing these ratios affects like binding capacity. So this is a diagram that basically covers the whole experimental procedure. And the starting tube on the top left is one of our 10 samples of xyloglucan and CNC. And each sample is left overnight in a homogenizer. And then 5 ml of sodium chloride is added. The sodium chloride will make the CNC unstable and help precipitate it in our next step, which is centrifuging it. And when it precipitates, the xyloglucan will bind to it and precipitate with it. So when we centrifuge it, as you can see, there's a pellet on the bottom. And in the pellet on the bottom, there's the xyloglucan and CNC, which has bound together. And in the supernatant, we just have the remaining xyloglucan. So in our experimental procedure, we do not need we only need the supernatant, and the pellet will be discarded. And in our pellet, supernatant, as I said, there will be the remaining xyloglucan, which will be used in our Orsinol reaction. So in our Orsinol reaction, we'll add 10 ml sulfuric acid, and then we'll also add the Orsinol. And this is left in the ice bath, which will create our cherry red color. So using our cherry red colored solution, we do not know what the concentrations of xyloglucan in there are. So in case we do need to know that, we need to pass it through a spectrophotometer. But when we pass it through a spectrophotometer, we need to first make a calibration curve using known concentrations. So this is an example of a calibration curve. Ours is later in a different slide, which I'll show you. But this will help us compare and measure the unknown concentrations in the red solution from the arsenal. And then we used the spectrophotometer to measure unknown concentrations at 425 nanometers. So here's a picture showing how the supernatant of each sample looks like after passing through the arsenal reaction. So as you can see, at the 25 concentration of CNC to xyloglucan, it's a much yellower color compared to like the 250. And this is because the color differences are depending on the xyloglucan concentration. So if there's more remaining xyloglucan left in the supernatant, the stronger the reaction will be, so the more red the color. So here's our calibration curve, as you can see on the graph to the left. And using this calibration curve, we were able to find out our remaining xyloglucan concentrations in our unknown red solutions. And then once we found the unknown concentration, we could plug it into the binding capacity equation, which gives us the amount. It gives us like a value for measuring like the binding between the CNC and the xyloglucan in the pellet. Talking about the graph, as you can see, as we increased in the X direction, which is increasing our concentration of xyloglucan to CNC, our binding capacity also increased until it hits like a plateau at around 200. And as we can see that it's approximately 271 xyloglucan over milligrams of CNC. And this is the maximum binding capacity, which was seen in the graph. And other papers also had a binding capacity of 98 or 250 or 333, and they have differing numbers due to it depends on the xyloglucan and the CNC where they extracted it from. For example, the Gu and Catchmark paper reported 98, but they used CNC that was extracted from cotton. So our CNC or our xyloglucan was used from tamarind, so it's all dependent on the source. We also observed that adding more xyloglucan beyond the binding capacity of CNC works as a viscosity enhancer. So this way you could easily like modify the viscosity of your solution. And it's a really helpful application in biomaterials. So first of all, I would like to thank my mentor, Ariane Fernandez, for guiding me and helping me explore this wonderful concept. I would also like to thank the Talaria team for this wonderful opportunity that has truly helped me become more confident in exploring scientific research. And thank you for listening to my presentation.